Many parts go into a modern video endoscope. Quality control is a key aspect, and every batch is checked using a computerized measuring machine. Measurements are compared to the design specification. Complete precision is essential, especially at the tip in view of its miniaturized components. Painstaking handwork also goes into the assembly of each individual endoscope. The production technologists are highly skilled. Precise hand skills are involved in soldering the small components. Constant quality control checks are made, and key parts of the assembly process are also done under microscope view. Here the lens optics are being assembled in a jig. The very complex control body and the angulation control wire adjustments need obsessional technique. Adjustment, lubrication and assembly are a highly personalized matter. Everything is done with high standards of cleanliness. Over the last 50 years, Olympus Tokyo has pioneered optical and digital technology, leading to major innovations in the field of endoscopy, like the gastro camera, the fiberscope, and the latest range of video endoscopes. Local agencies, such as Olympus Keymed in the UK, play their role distributing the latest range of endoscopes, and they're responsible for servicing and repair. All digital cameras are based on a CCD or charge-coupled device. A CCD consists of an array, tiny rows and columns, of small cells known as pixels. A pixel is a picture element which converts light into an electrical charge. The CCD of a conventional camera is miniaturized for video endoscopes. Here is the CCD camera of a video colonoscope. This is the lens assembly. There are two kinds of CCDs and processors around the world. The commonest is the white light system, known as the 100 series of Olympus. For this, the CCD pixels are overlaid with tiny colour filters. Some pixels receive only blue light, some red light and some green. By this means the white light CCD outputs a colour image directly. Japan, Britain and some other countries use the alternative 200 series sequential or RGB system, in which a rotating filter wheel in the light source produces alternating flashes of primary colour light. These primary colours are summed 5 times a second in the processor, resulting in true colour on the monitor, but with 3 times the resolution, because all CCD pixels are used for each image. However, rapid movement or bubbles can cause colour breakup, and the primary colour flashes, sometimes seen using 200 series instruments. This is the bending section of a colonoscope. Inside the bending section there are 4 pull wires. They work a bit like the reins of a horse. Pulling on one wire angulates in the chosen direction. The insertion tube, or shaft, is complex and tightly packed. Inside, as well as the electrical connections, are 2 light bundles, air and water channels, the biopsy and suction channel, and the 4 angulation wires. These components need to be correctly ordered and lubricated to ensure that they move freely. The outer waterproof plastic layer covers a stainless steel braid. But critically, inside this, there are 2 phosphor bronze spirals, one clockwise, the other counterclockwise, which together give the shaft its torque stability. The components running through it terminate at the light guide plug with its waterproof cleaning cap. The light guide plug contains, as well as the suction and water bottle connections, the metal probe covering the light guide, with the fibre bundle inside it, and the air channel probe. Finally, the pin unit, if removed, covers 3 sets of wires, from the CCD, the control switches, and the light control system.